Hi everyone, thank you all for coming today. It's a pleasure to be here. My name is Niki Moya, and I am a PhD candidate in the Neuroscience department here at Northwestern. And when I came here, I took a particular interest in studying the neurobiology of schizophrenia. So that is what my presentation is going to be about today. And to first start with a bit of background information, schizophrenia really is one of the most devastating mental health disorders to date, currently affecting about 20 million people worldwide. And once diagnosed, patients with schizophrenia have a significantly higher risk of premature mortality with a reduced life expectancy of about 10 to 20 years on average. And unfortunately, a main contributor to this is an increased risk of suicide. Patients with schizophrenia also experience significantly higher unemployment rates, with about 60 to 80% of those individuals with schizophrenia being unemployed. This, on top of healthcare costs, contributes to a large economic burden. Schizophrenia is also still highly stigmatized and misunderstood even to this day, which contributes to barriers in treatments. It's estimated that about 50% of individuals with schizophrenia do not receive proper treatments, and even in those that do, these therapeutics don't comprehensively address all symptoms. So because schizophrenia poses such a major public health issue, it's really important to understand the neurobiological mechanisms that drive schizophrenia in hopes to treat it. Now, in the schizophrenia field, there is a popular hypothesis known as the dopamine hypothesis of schizophrenia. And the dopamine hypothesis states that in patients with this disorder, they have increased dopamine release specifically in one area of the brain. And this increase in dopamine contributes to schizophrenia symptoms. But what are the consequences of having too much dopamine release in the brain? Dopamine is typically thought of as a good thing, right? Well, just like too much of anything, too much dopamine can actually be bad for you. And this is because an overflow of dopamine in the brain can change neural activity. And this mainly happens through dopamine receptors, or the receptors on neurons in which dopamine binds. There are two main types of dopamine receptors, and these include the D1 and D2 dopamine receptors. And when patients with schizophrenia have increased dopamine release, this dopamine binds to dopamine receptors, causing them to be hyperactive. And this hyperactivity can change neural activity and behavior. Now, to combat this, current antipsychotic drugs mainly target the D2 receptor and are classified as D2 antagonists. So they are meant to block this increase in dopamine from binding to D2 to hopefully alleviate some schizophrenia symptoms. And these antipsychotic drugs are effective in treating symptoms you'd typically think of when referring to the disorder. So things like hallucinations, delusions, and thought disorders. However, many patients with schizophrenia also experience these symptoms called negative and cognitive symptoms. And these symptoms include things like loss of emotion and social withdrawal on the negative symptom side, and then also deficits in executive function and working memory and attention on the cognitive symptom side. And current antipsychotic drugs actually lack efficacy in treating these negative and cognitive symptoms. But why is that? If increases in dopamine are supposedly involved in schizophrenia, shouldn't antipsychotic drugs that target dopamine receptors work for these symptoms as well? Well, these antipsychotic drugs don't do a good job at targeting that D1 receptor, the other main dopamine receptor subtype found in the brain. Because of this, changes in D1 activity could contribute to these treatment-resistant negative and cognitive symptoms. However, this idea has never been explicitly tested. So the role of D1 hyperactivity in negative and cognitive symptoms remains unclear. Because of this, my hypothesis is that treatment-resistant symptoms are caused by dopamine-driven changes in D1 receptor activity rather than D2. So that is what my project aims to explore. But how do we begin about addressing this question? It really isn't easy to see what different neurons and receptors are doing in the human brain. Well, our lab uses mouse models. And we do this by increasing dopamine release in the same region of the mouse brain where patients with schizophrenia experience, which essentially mimics that neuropathology seen in schizophrenia. I can then visualize how this increase in dopamine is changing D1 activity by using different research tools. And the research tool I use to visualize D1 neurons is called GCamp. GCamp basically works by making neurons light up when they're active. So I can inject GCamp directly into the mouse's brain, and once there, GCamp embeds itself into the cell membrane. When neurons become active, calcium ions come into the cell and bind to GCamp. And once calcium is bound, GCamp emits this green fluorescent protein, which makes these neurons glow when they're active. I can then visualize how increases in dopamine are changing D1 activity by mounting a miniaturized microscope on top of the mouse's cranium. And I can record and measure neural activity while this mouse is freely moving and behaving. And I can do this because this mini microscope doesn't weigh too much, so it doesn't impede the mouse's natural behavior. And here are some example videos of what that kind of looks like while a mouse is initially freely moving in an open field, where I can compare normal resting states to hyperdopamine schizophrenia states to see how increases in dopamine are changing D1 receptor activity. I can also do this during social and cognitive behavioral tasks, which are proxies for negative and cognitive symptoms of schizophrenia. So by defining the role of D1 receptor hyperactivity in the neuropathology of schizophrenia, my hope is that we can develop new therapeutics that more comprehensively address all symptoms of the disorder. Thank you.